In an earlier segment, we learned that digestion occurs via digestion enzymes that catalyze the breakage of peptide bonds in proteins to ultimately release individual amino acids. One question that comes to mind is, why is it possible that these digestive enzymes don't run wild and just decompose all of the various proteins within our body? We'll answer that question here and others as we look at how enzymes are controlled through covalent modifications. Covalent modifications are an alternative route to controlling enzyme activity to the allosteric regulation that we learned about in the last video. Enzymes, being proteins, are formed by the creation of peptide bonds, also known as amide linkages, between individual amino acids, so that ultimately hundreds or even thousands of amino acids are linked together and fold into a specific three-dimensional shape that has catalytic activity. This is not the end of the story. In addition to that peptide chain, there can also be covalent modifications occur of those enzymes to control their activity. Covalent modifications are where groups are added or removed from a protein's amino acid chain in order to impact the activity of the protein. There are a bunch of different types of covalent modifications, too many to cover in an introductory topics course. We'll talk here about two common covalent modifications that will give you a general idea about the flavor of these modifications and their impact on the activity of enzymes and why these are a really powerful way for the body to control the activity of enzymes. Two common types of covalent modifications are Zymogens, which we'll spell out here, also known as proenzymes. Pro means pre or before. So Zymogens or proenzymes refer to enzymes that are produced by the body in an inactive form initially. Usually, this results from a section of the polypeptide covering the active site, and then later on, the proenzyme is converted into an active form, usually by a section of the polypeptide chain being cleaved off to unveil the active site. The second main type of covalent modification is referred to as phosphorylation. Phosphorylation is where a small group known as a phosphate group will bond, to a protein at a specific location, and the addition or removal of a covalent bond to that phosphate group will activate or deactivate the protein. And in fact, phosphorylation is a very, very common modification to proteins. In fact, about a third of human proteins rely upon either being phosphorylated or dephosphorylated to be active. We'll spend the rest of this video talking in more detail about why Zymogens are important and how they work, as well as looking at phosphorylation and how that works to control enzymes. We'll also explore why these modifications are important in controlling exactly when and where enzymes act within the body, so that things like digestive enzymes don't end up eating us alive and instead remain active only within the digestive system. Let's talk about Zymogens, also known as pro or pre-enzymes. These are enzymes that are produced in an inactive form and then later activated via the removal of a section of the enzyme. How does this take place and why is this important? Let's look at this schematically. We'll start with this diagram where we begin with a zymogen. The zymogen is this entire protein here, where we have this blue part labeled as the inactive state of the enzyme, and you'll notice that it has this appendage covalently bonded to it. This is two parts of the polypeptide chain. What happens is that the zymogen is generally produced in one location of the body, where it remains in its inactive state, and then once it reaches a specific location of the body where it needs to be active, there is waiting for it at that location a so-called activating protease. Remember that proteases break bonds within peptides. That's a very specialized protease that breaks the covalent bond here between the inactive state of the enzyme and this subunit shown in orange here. Once that breaks apart, the enzyme is then able to form its active state and go about functioning and catalyzing reactions. There are a variety of examples of zymogens and their role in real life. One example of zymogens playing a role in real life is in the process of blood clotting. When you get a cut, it is important that enzymes work quickly to allow that cut to heal by forming a blood clot as one of the first steps in that healing process. However, if blood clotting ran wild and clots were forming in undesirable locations within the body, a variety of cardiovascular symptoms would result, and that would be a very negative thing rather than a positive thing. As a result, it is essential that blood clotting be carefully controlled by the body. The way that this happens, or one of the ways that this is controlled, is through Zymogens. One of the blood clotting enzymes is produced as a Zymogen hanging out within the body, and once a cut is detected, a series of responses is triggered, one of which results in that enzyme being converted from a Zymogen into an active protein by cleaving off one end of the protein so that the active site of the protein is revealed and the enzyme can then act in clotting blood. In addition to Zymogens playing a major role in blood clotting, they are also essential to digestion. You will notice here in looking at this chart in the lower left corner that we have listed on the left the Zymogen, which is the inactive form of the enzyme. That inactive form of the enzyme is produced in one location. You'll notice here a variety of places, including the pancreas and liver, as prominent places where digestive enzymes are produced, but those aren't ultimately where it's desirable for those enzymes to work. So what happens is those enzymes are produced and stored in those locations, and then they are transported to their final destination in the stomach or the small intestine, usually, where they become the active form, because it is in these locations where the enzymes are activated that proteases exist to cleave the bond between the inactive form of the enzyme and its repressor to ultimately release the active digestive enzyme that we list over here, and these have the common names that we recognize as the names of these different digestive enzymes. So what happens then in instances where an inactive enzyme, a Zymogen in other words, becomes activated in the location where it was produced rather than first traveling to the location where activation is desired? The effects of activation of these Zymogens in inappropriate times or inappropriate locations can in some cases be deadly and cause a variety of serious diseases. For example, one cause of pancreatitis, an inflammation of the pancreas that can be life-threatening, is due to the activation of Zymogens that are produced in the pancreas but should be transported to the intestine or to the stomach before they are actually activated. If those digestive enzymes are activated instead in the pancreas, they can cause a severe inflammatory response and essentially result in those digestive enzymes to some degree digesting the pancreas, which is definitely not the desired effect and definitely has some serious health consequences. In addition to Zymogens, as an example of covalent modifications that control the activity of enzymes, we also look at phosphorylation as a very critical way that the activity of enzymes is controlled. In fact, it is estimated that at least a third of human proteins are controlled by phosphorylation reactions. What happens in phosphorylation reactions is that phosphate groups, PO3, are added or removed from specific residues of the enzyme, and by adding or removing phosphate, that impacts the activity of the enzyme by either changing the three-dimensional shape of the enzyme and therefore impacting how the enzyme binds to its substrate at the active site, or by the phosphate group directly engaging at the active site and interfering or activating the binding of the substrate to the active site. So how are enzymes phosphorylated, and where does the phosphate group come from anyway? Let's begin this discussion by taking a look at the structure of ATP, adenosine triphosphate. ATP is the source of the phosphate group. What happens is that when an enzyme is phosphorylated, this PO3 group is lost from the terminal end of ATP. When ATP loses a phosphate group, we call that phosphate group PI for inorganic phosphate because there are no carbon atoms associated with it. The product of that is ADP, adenosine diphosphate, because the structure no longer has that third phosphate group. Instead, it just has the two phosphate groups here, making it ADP instead of ATP. ATP doesn't randomly and spontaneously donate a phosphate group to enzymes during phosphorylation. Instead, that process itself is controlled by an enzyme. What happens in the process of phosphorylation is if we take an enzyme, I will go ahead and draw this lovely enzyme here. We will call this our inactive enzyme. And our inactive enzyme comes into contact with a so-called kinase. A kinase is a type of enzyme that catalyzes the transfer of a phosphate group from ATP onto the enzyme. And so what happens during this reaction is that the kinase binds to both the enzyme that it is phosphorylating and to ATP, adenosine triphosphate. And during this reaction, it forces the reaction of ATP with the inactive enzyme to result in the transfer of the phosphate group from ATP onto a specific location of the enzyme. It doesn't just transfer it randomly anywhere on the enzyme. Instead, it transfers it onto a specific site. So I'm going to draw an arm coming off of that, a covalent bond, linked to a phosphate group, PO3. And with that bonding at a specific site, what happens is some structural change of the enzyme, such as carving out an active site here, as a result of adding that PO3 group. Hence, the enzyme goes from becoming inactive over here on the left to, once it's been phosphorylated on the right, being an active enzyme. The other product of this reaction results from breakage of a phosphate group from ATP. When we break off a phosphate group from ATP, as we saw up here, ATP becomes ADP, going from adenosine triphosphate to adenosine diphosphate. So I will go ahead and put also on the product side of the equation ADP here. Now we've just looked here at a case where phosphorylation results in creating an enzyme that is active. There are also scenarios where dephosphorylation, the removal of a phosphate group from an enzyme, is required in order to activate the enzyme. This is very specific on a case-by-case basis for different enzymes. Let's take a look at that process of going from a phosphorylated enzyme that is inactive to a dephosphorylated enzyme that is active. We start here with an inactive phosphorylated enzyme. We know it's phosphorylated because we see that PO3 group covalently bonded to the enzyme. There are specialized phosphatase enzymes, where phosphatases are responsible for the removal of phosphate groups. And when a specialized phosphatase comes in that recognizes this particular enzyme, what happens is that the phosphatase uses water as a reactant. So I'll go ahead and put plus water here. And when water and our enzyme with the phosphate group binds to the active site of the phosphatase catalyzes the breakage of this covalent bond sliced here in order to remove that phosphate group and leave behind the standard R group from one of the 20 amino acids. This, as a result, creates an enzyme product that, once that phosphate group has broken away, experiences a conformational change, meaning a change in shape that results in the enzyme becoming active, as we can see here from the fact that the product of this, the dephosphorylated active enzyme on the right, now has an active site shape there that a substrate can bind to and ultimately undergo a reaction. So what we've seen here is that phosphorylation is a key driver of enzyme activity and a key way that the body regulates whether an enzyme is active or inactive. On a case-by-case basis, some enzymes require phosphate groups in order to be active. That installation of the phosphate group is catalyzed by a kinase. In other enzymes, instead, the phosphate group prevents activity of the enzyme. In those cases, a phosphatase is needed to remove the phosphate group and create an active dephosphorylated enzyme.